All right, good morning, everybody. To me, it feels like a Monday, but no, it's not. We're halfway through this week. That's even better. Any questions or concerns before I start with stuff today? All right, so this chapter I think is interesting to start thinking a little more, some more details as we move into thinking about microbes. We want to think about some of the tools, some of the techniques that we utilize to visualize organisms. And there are all sorts of different types of microscopes. We will focus in here, and although we're going to show you a bunch of other stuff, we will really focus on compound microscopes, light microscopes, which is what you're going to be thinking about when you deal with stuff in the labs here. We will show you some other potentials, and oftentimes as we're progressing through the chapters, we'll see some images, and they use lots of electron micrographs. So electron microscopes to zoom way in on microbes and give us some details, all right? I told you before, oftentimes seeing stuff like bacteria under just a regular microscope is really not very impressive. Little specks that, you might think of, you're seeing, purple colored dust or something like that. It's very, very small. But when you use an electron microscope, you can zoom way in. And now these things that are maybe not so impressive, at least to me, all of a sudden are super cool as we see some features that you definitely don't get with just a regular microscope, right? And it really kind of depends on the circumstances as far as what you might be needing, all right? Doing just regular microbiology, if you think in a clinical situation and a patient comes in and we cultivate these organisms, we want to stain them up and see what's going on. We want to see, are they gram positives or negatives, and some details about the morphology. But we don't bust out the electron microscope and zoom way in on it. That is way too much detail, too much information, and too costly, all right? And time consuming as well, all right? If we're doing research, well, that's a different story. And maybe we want some, nice high resolution images of the surface of whatever cell we're looking at, or the interior of them as well, all right? There's numerous potentials. And other microscopes that might show us things happening in real time, or other possibilities. Not that our compound microscope can't give us some live specimens and the ability to visualize those things. There's others that we can potentially employ as well. So this chapter, really, we think about all sorts of different microscopy techniques, right? That's really the idea. And there are numerous things, again, that we will try to show as we progress through here. I don't want to get bogged down and start talking about specific microbes and features and things like that. This is really more of just kind of a quick show and tell of the different types of machines, right? Although there are a few that'll give you some extra stuff, all right? So here we've got a couple of images, and these are things that we think about with your regular microscopes, the bright field or compound microscopes, a couple of different potentials, all right? The dark field is this next image, and it is something that we can employ with our regular microscopes, the compound microscope, all right? So it can give us either bright or dark, dependent upon ways that we switch some stuff up with the microscopes. When we're dealing with stuff in the lab, it's always going to be the bright field as we visualize these things, using light to focus in on our respective organisms, all right? So a couple of different things in here. This image, again, a bright field microscopy. This is, I've mentioned a little bit about gram staining, and we're going to show you more details with this. This is a gram stained image where we're able to differentiate between types of cells, positives or negatives, and basically what bacteria have thick, we would call them gram positives, thick cell walls, or very thin cell walls, and those are the gram negatives, all right? So that gives us some potentials to differentiate and think about them on different angles as we're evaluating the types of microbes that we're examining. So this image has some other stuff going on in here. There's lots of other cells that we can see. And hopefully, as we look at this, you can pick out some differences between our bacterial cells and the others, right? These are types of eukaryotic cells. Nuclei are very visible, right? And really what we're seeing here are a bunch of white blood cells. Highly likely that the bulk of these guys, and this is, again, a gram stain. It's not a blood smear where we're staining things up. That might allow us better differentiation of the types of white blood cells that we're seeing, right? They would look a little bit differently based off of a blood staining procedure rather than a gram staining procedure. That matters not, right? There are numerous white blood cells in here, and my gut tells me these guys are all neutrophils. We'll see some details of this later on as we start thinking some of the immunology. But they're some of the most common white blood cells that you would see in a smear in the first place. So it's highly likely that the bulk of what you're going to be visualizing would be those guys. And then some other, probably lymphocytes, T cells or B cells, something like that, all right? You can tell seeing these immune cells, human cells, these eukaryotic cells with their nuclei compared to a bacterial cell like this guy, these are all kind of linked together, maybe 3 of them together. But here's a single one, here's another single one. The bacterial cells are considerably smaller than the eukaryotic cells, right? About 10 times smaller, right? And you don't really see lots of details with the bacterial cells because there aren't a lot of details to be showing in the first place, right? They don't have organelles and membrane-bound nuclei and all of that stuff that we can potentially see with the eukaryotic cell, all right? So light microscopes give us opportunities to visualize things, bacteria or eukaryotic cells, and make some, draw some conclusions with that. This tells us right here, we're looking at anthrax, Bacillus anthracis, but you can't look at a gram stain and immediately peek in there and say, oh, that's anthrax, right? I can tell that just by how it looks, right? That's not what we're getting out of it. There are numerous types of bacteria that will have similar morphology, right? It definitely tells us some things. I already mentioned gram positive. It tells us some features about the cell, and we can also see the morphology that these guys are bacilli shaped or rod shaped, right? So we've got some ideas, right? And knowing that, we could in turn try to, if we wanted, we could try to cultivate these guys, and we know we're looking at a gram positive. Maybe I could put it on this type of plate, and that would help it to grow. or other things like that. And we'll talk about some of those details later on. Like, how do we get these guys to grow in the first place, right? The next image over here, this dark field shows us kind of a situation where the bacteria, instead of them being stained up, the light is like shining right through them. The slide itself has been stained and the organism not, right? And so the light basically goes through there. It kind of makes me think it's not, but it's sort of like glowing in the dark. We will show some fluorescence microscopy that literally is glowing in the dark. These guys aren't glowing in the dark, but as the light, because it's still a light microscope, as that's shining up under the slide, the organisms did not pick up any stain, only the slide was stained, and that in turn gives us this sort of glowing type of an image. So this is the organism that causes Lyme disease, Borrelia burgdorferi, and spirochetes, so corkscrew-looking bacteria. And you can see pretty easily comparing the two, morphologically, they are different, right? You also can't tell just based off of this that we're looking at Borrelia, right? We know we're looking at a spirochete, but that's all you get from that. So there are a few details in here that we need to at least think about the littlest bit when we're trying to understand how we visualize organisms in the first place. How is it that we're able to capture these images? So when we think about this, we need to think about light as wavelengths. And with that said, the distance between these wavelengths are differing amounts of energy. And that's what we can see over here. Lower frequency, higher frequency, and we'll see the visible spectrum here in just a second of what we're able to perceive, right? So with that, there are also other things we need to take into account as we're trying to visualize things. Our Petri dish over here with our little bit of media in there, the red stuff is the media. The Petri dish is transparent, plastic, although back in the day they were made out of glass. Easy to look right through and see organisms growing versus this chunk of meteorite that is opaque, where light would not transmit through it at all, right? The slides that we put our microbes on, the little chunk of glass allows the light to pass directly through that, but there are some potential issues with that. And we need to ensure when we're trying to look at bacteria that we're maximizing the amount of light that we're able to harness or focus in on, and we don't lose some of it, all right? And we'll see that here in just a second, all right? So refraction, and this is our little chunk of glass. Refraction happens as light passes from one medium or substance into or through another, all right? And when it comes to trying to visualize organisms, we want to try to minimize as much loss of light as we possibly can, all right? So we'll want to take that into account, and we'll show you some details of this here in just a second, all right? And that you don't need to get bogged down with this, the refracted and reflected light rays, but ultimately we try to minimize these issues, especially with the reflection, right? We know refraction is going to happen as we're moving through there. That's a potential issue, right? This is a good image that shows some of the refraction. So we've got a metal rod going down into this water, and it looks as if as it hits the water that this pole is bent. But it is not, right? We should have a side by side so you could see it's really not, right? But it appears as if that's the case, all right? And it has to do with the refractive index of air versus water. They are different, and as this happens, it makes it look a little different. It's the same as this, right? it's going in, but it looks as if it has bent, all right? So, and you know, as soon as I opened this chapter up, I already got into the classroom and I thought, geez, I should have brought in a bottle of immersion oil, but that requires me to run back to the building and go to the basement and rummage through all of the microscope staining trays to find the right bottle, right? There are, and it's highly likely in the intro lab, you guys will have some immersion oil that we will use for our staining. Some of these will be in little glass bottles. Others are in plastic kind of droppers. That's not as impressive, right? The glass bottles, there is some interesting stuff. And I was never a fan of physics. There's lots of stuff that I'm just like, I don't want to think about that, right? I can tell you oftentimes as these chapters open up, instead of saying microbiology right at the top, the first thing it says is physics, and it gives me just a little bit of panic attack. I'm like, oh no, what is this? And where am I, right? I hope I'm not in physics class. This really is dealing with some of that stuff. And one thing that I recalled was this, and it really was because I could visualize it with some microbiology stuff. And I was like, wait a minute, that makes sense, right? So with this metal rod, all right, that oil that we will use, and I'll show you this in just a slide or two down the road, when we are looking at things under the microscope, bacteria under the microscope, we need to zoom way in on these organisms, and we use a drop of oil that we put onto the slide, and that basically goes up into the lens and it lets the light shine right through, and we don't get a bunch of reflection with that light as it shines through the slide, right? Little bit of refraction, but the reflection, we minimize that dramatically. The oil and the glass slide have the same refractive index, right? So it behaves as if it's the same thing, the same type of medium, and the light just moves right up into our lens, right? That's what we want, right? Those little glass dropper bottles of immersion oil, as you unscrew the lid, they have a glass rod that will have a drop of oil on it that you touch onto your slide, all right? Some of these dropper bottles, nice and clear glass, as you go to put the dropper back into the oil, you can see the glass rod, you can see it plain as day. And as soon as it goes into the jar full of oil, you no longer see it at all, right? It's invisible. It looks as if you maybe broke it, right? It's completely gone until you pull it back out. It's stinking magic, right? It's because it's the same refractive index, and that glass goes away once it touches the oil, all right? You don't see it bending. You don't see it at all right? That is awesome, right? And that's what I remembered as I was learning about this type of stuff and wavelengths and refractive indices in my physics class. All I remembered was the glass rod disappears, right? Which wavelength is high powered and low powered, right? All of this, I'm like, I don't care, right? That matters not. I just want to see the physics teacher, you know, have some student lay down on a bed of nails and crush a cinder block. That was awesome, right? That's what I remembered. Not all of, not, you know, velocity and acceleration and whatever. That's why I'm here, right? And not down teaching physics. So here we could see some details with this, and it depends on your circumstances. A prism, all of these different types of lenses, right? And so this prism will basically take the visible spectrum and separate it into the respective wavelengths that you can see, all right? So we've got a very high refractive index, and the light as it moves through this prism is separated, right? That's kind of cool as well. There's different types of lenses, and this kind of makes me think of like funhouse mirrors, sort of similar idea as we think about this, how it focuses the respective images. And what we utilize in microscopes allows the image that we are seeing to be zoomed in and focused on And it's the result of a handful of different lenses focusing that image. It's not just one, all right? So with our convex, we've got our light, and both of these, it's light passing through this direction. So light passing through to a focal point, something at this point here that we can visualize. If we are out of focus one way or the other, you lose resolution. It becomes very blurry and out of focus, right? The focal point is where we are able to discern whatever image we are trying to see. In this circumstance with the concave, it's the opposite, with the focal point being before that. Okay. So this electromagnetic spectrum, this is back to those wavelengths that we showed just a second ago. The visible bit is just a small fraction of the electromagnetic spectrum. What we can actually see, what we can perceive, all right? There is a tremendous amount on either end that we are unable to visualize. Doesn't mean we can't necessarily perceive it. Yes?
So for animals that can see more colors than we can, does that just mean we can see more electromagnetic frequencies?
For sure. So yeah, they can pick up, they can see some other stuff. They're just picking up a little bit extra within here, a little bit more UV or infrared, kind of depends, right? Depends on the animal, right? I think goldfish are supposed to be able to see more infrared type of stuff. I don't know, right? We had a goldfish and I'd always try to shoot the remote at it just to see, does he know, right? He didn't care, right? So I don't know. That's not truly a scientific study, right? It's just changing channels. And I'm like, what do you think, goldfish? He didn't care, right? I don't know. UV stuff, I think things like bees, I think are supposed to be able to be perceiving things more in the ultraviolet. So yeah, they're able to pick up different things, right? I saw this thing, I didn't click on it. I don't know if it's true, but it said, oh, this is how your cat sees you. And it was a person and they were all striped. And I'm like, oh, do I have stripes? I wonder, right? What does the cat think? I don't have a cat anymore, right? So who knows, right? It's hard to say, right? Do I have stripes? It is interesting, all right? So we could see as we go down or up, so higher energy, those wavelengths would be super close together, almost packed side by side as we get high energy, and very spread out as we get to lower energy, right? And it depends again on the circumstances. So and we're not going to go through details with all of that stuff. So like I said, there are a lot of images that we move through this chapter to kind of just show you different types of techniques or tools that we can utilize to visualize things, all right? So this uses some specialized dyes, some specialized staining techniques with dyes that when exposed to ultraviolet light, they will get excited and fluoresce, so glow in the dark, all right? They don't just, not just by themselves, but the UV light helps to get that to happen, all right? There are all sorts of potentials with fluorescence, and different things may fluoresce or may not when exposed to a black light or UV light. You may see some of these things glowing, right? So this does require some specialized techniques, special staining procedures, and specialized microscopes. It's not something that we can just flip onto our compound microscope and expect to see, right? There are all sorts of things that we may try to focus in on or think about with these types of fluorescence microscopy. Different types of stains will pick up different molecules. We may be staining for lipid or proteins or nucleic acids. There's numerous substances that will pick up different types of dyes and in turn give us a perspective of whatever sorts of cells we're looking at. So nuclei here in blue or purple, and we've got other proteins, structures, and maybe, I don't know what the red stuff is. I don't know if it's telling me all of these things or not. Microtubules, so proteins, the nuclei, and actin filaments. So other actin and microtubules are different types of proteins, but the stains link up with them specifically, and that gives us some details about the features of these cells. So this a bovine pulmonary artery cell, endothelial cell, we could potentially be visualizing these, and we've got a handful of these cells. We may have the opportunity to see some that are in the process of replication, where the membrane of the nucleus has been degraded, where microtubules are in the process of pulling them apart. Fluorescence microscopy can lend to some of those features where we can visualize things. Most of you that took biology probably looked at the root tips. You weren't seeing details like this, but if we had stained them up with these types of stains, we would have, right? And we would see green and red as we are linking up and trying to grab those chromosomes. And if we had stained with some DNA stain, you could see those chromosomes glowing in the dark as well, right? All in a nice line with metaphase or in anaphase as they're being pulled apart. We could see all of that stuff. And there's images, and definitely they show in biology books that give you some fluorescence microscopy to see some of that process. And I think that's pretty stinking cool. With bacteria, there's also other potentials where we may be staining things up in different types of cells, picking up different colors and then visualizing them. It's not going to look maybe as massive because, again, bacteria are considerably smaller, but you may have a wide assortment of different potentials of cells. So we've got to think a little bit about microscopy and back it up to get some historical perspectives to hopefully shed some light and really make you think a little bit about the microscopes that we're going to be working with in the first place. I feel like we take a lot of stuff for granted as we get in there and we're preparing a gram stain or something, you know, something like that, looking at something under the microscope. It is something that, you know, you might be doing repeatedly and you don't even really think anything of it, all right? It depends, all right? Our first image over there is Antoine van Leeuwenhoek. We think of him as really one of the initial individuals that started looking at microbes, like a father of microbiology, father of microscopy, right? He looked at all sorts of things and really pretty impressive. There's all sorts of details about Leeuwenhoek that I think are really pretty fascinating, right? He was not a scientist, right? But he definitely was a scientist, right? It seems very odd. He was a merchant, a shopkeeper. He was selling all sorts of things like linens. And it was important to know that what they were saying was what it actually was, right? He wanted to look at the linen. He wanted to look at the threads, the thread count, right? He was the first concerned about thread counts in sheets, right? He wanted to know, right? Is this super fine linen or is this some cheap knockoff, right? What are you trying to sell me here, right? That's really kind of what it was all about. So this is a microscope that he built. It looks nothing like microscopes like we think about today, right? Very odd, very simple, right? It didn't allow him to get super zoomed in images to see bacteria, but he could definitely see all sorts of other things and the hints of some bacteria, all right? But not like the, you know, seeing them under a regular microscope. They could magnify, I think, 300-400 times, all right? Which is pretty good, but to get nice images of bacteria, again, not impressive, but nice images, it's about 1,000 times magnification, right? He can see him at 400, but not so well, right? So he looked at all sorts of things, and it's really impressive to me. And again, stuff that I would say we kind of take things for granted. We slap something on a glass slide, we look at it, and we get rid of it, right? We put it in the container. They technically wash those slides and reuse them, but they wash it off. They just get rid of it. We don't care about it, all right? Leeuwenhoek didn't think like that. He wanted to see stuff and he wanted to go back and look at it again, all right? Every time he wanted to see something, he built himself a microscope and he visualized it. And he had a shelf that was full of microscopes, hundreds of microscopes that he built. And every single one of them had a specimen that was fixed on there that he could hold up and he could visualize. He could go back and look at it, right? A whole collection of microbes, right? And he saw all sorts of stuff, all right? Like I said, he's a merchant. He's not a scientist, all right? But he was. He looked at these things and he drew pictures. He wrote great descriptions about what he saw. His pictures were fantastic. As he draws these things, he starts submitting them to the Royal Society. They're looking at stuff. They're like, they can't believe it, that he's able to see these things. All sorts of people start paying him visits to look through his microscopes. The Queen of England comes to see some of his wares. She probably left with some sheets, right? But she wanted to see what sorts of stuff he had found, all right? He looked at all sorts of stuff and saw these animalcules from droplets of rain with protozoa that he was able to visualize, or wee little beasties, these crazy, again, protists for the most part, that he was able to see. Any sorts of bodily fluids and secretions he looked at, his or others, scraped plaque from his teeth and saw all sorts of things, probably the beasties, right? All sorts of microbes within there. He described Giardia, which is an intestinal protozoal parasite. He collected his own diuretic stool, slapped it on a microscope and looked at it, all right? And drew some pretty impressive pictures, all right? All sorts of stuff, all right, that he was able to see and describe. And it sparked interest in the scientific community. They wanted to know, right? What are these things, right? Impressive, right? I kind of, you know, it is mind-boggling to me to think about this. So as he's building these microscopes, he's grinding the lenses. He's taking pieces of glass and grinding them down to give him the ability to zoom in and visualize these things. Very, very tedious work, very patient person, right? As he grinds these lenses and is able to visualize these things and draw these images, the magnification and the quality of the lens that he was able to produce couldn't be replicated, right? Nobody else was able to make a microscope that was as powerful and provided images so clear as Leeuwenhoek, right? It took almost a century before there were others that were able to see things like he was able to see, all right? Which makes me back it up and say he must have been a time traveler, right? It makes perfect sense, right? He already showed up with these little lenses. He already had them, right? That's not true. But it sure makes you wonder, right? What's going on with this guy and how was he able to do that stuff, right? So Galileo, famous for telescope, among other things, right? But he was also a pioneer with microscopy as well. And he was doing similar things, grinding glass and visualizing things. But again, Leeuwenhoek definitely blew him out of the water when it came to the production of those specific lenses, right? A little bit later, Robert Hooke starts looking at organisms, and this is maybe a little more microscope looking than this guy, right? Little bit, right? So Hook, you know, I said a little later, they're near same time frame, really. Starts visualizing things, and in this case, it is a light microscope. It's different than what we think about today, but it's got definitely some of the same sorts of features, and it allows for the light to be focused in on the specimen, in this case, candlelight, or an oil lamp, maybe. That looks pretty fancy. And in this case, what we're looking at over here are some cross sections, some little slivers or shavings from the bark of a cork oak tree. And it was these first images that allowed Hook to visualize plant cells and coined the term cell in the first place, right? He is the one that came up with the term cell. As he starts seeing all of the cell walls of these plants, he envisioned like the small rooms or dormitories that monks lived in, and they were called cellulae, right? The dorms, the cells that they lived in, and that in turn is the name that we know. This is another interesting one. Zacharias Janssen, along with his father Hans, may have invented the telescope, the simple microscope, and compound microscope during the late 1500s or early 1600s, right? Although some of this is inconclusive, all right? So right around same time frame as Leeuwenhoek, all right? And definitely trying to produce similar sorts of images as we start thinking about moving into the visualizing this realm of the unknown. I do think super impressive as we see this name going clear back to the early 1600s. Janssen lenses are still highly prized and sought after. There are numerous microscopes that still employ these types of lenses. I think that is pretty cool, right? We've been in business for centuries, right? That's pretty cool. All right. So here is something that we will be more familiar with as we get down into the lab. Some of you probably already had a little bit of experience with some compound microscopes. So this is a typical bright field compound microscope. This is something that you will see in the lab, and you'll have some opportunities to look at some organisms. We're not going to spend huge amounts of time looking at things through the microscope, right? There are other labs where there's more focus with that, but for the introductory purpose, we see them a little bit, all right? Quickly, some details, some anatomy of the microscope. These use a light to illuminate instead of candlelight, right? So we've got our illuminator down here. There are numerous things, a switch to turn the power on and off. Typically, I don't know if you see that on here at all. The rheostat sometimes is also the light switch where you turn it all the way down and it clicks it off, and then you turn it, and based on how you turn, adjusts the intensity of the light, super bright or pretty dim, all right? There are others where it's separate, a switch and then a light rheostat adjustment, all right? You do want to be familiarized with some of these things because as you are spending some time looking through the microscope, you want to make sure you know how to make it work to the best of your abilities. You don't want it always to be super bright, but you also don't want it to always be dim, all right? You start off when you're trying to see stuff, you're giving a bigger view, it's less magnified, and you want that to be more dim because otherwise it's too bright. And as you get zoomed into the highest magnification, then you want it to be pretty much as bright as you can get it because you're only focusing a small amount of light. And if it's dim, you're not capturing much of that image at all, right? So the rheostat allows you to, adjust the intensity of that light. They've got everything all numbered up in here. I'm not going through the numbers like I should. That probably doesn't matter. There are knobs here. This is the stage where your slide will go, and there's a mechanism that holds it in place. These knobs move the stage, the platform, and they move the slide on that platform horizontally or vertically, right? The focusing knob, number 4 and 5, coarse and fine, moves the stage up and down, right? And that's what's going to allow us to focus. So we get up and down with the big knobs, and the smaller knobs on the stage moves the slide back and forth and giving you the ability to scan all over on that slide. You focus in on the lowest power with your objective lenses right here. They're all numbered, color-coded. The small stubby guy is the lowest power. The big one is the highest power. You always start with the smallest lens, all right? So you use the coarse focus, the big knob, to get that specimen slightly into focus. It's not going to look great on that low power, but you see the hint of something, all right? It might be the hint of some stain. You may have drawn on your slide with some wax pencil, and you see the wax pencil. That's a great way when you're first doing microscopy. Have something that's huge and easy to see, all right? And you see this huge black line, you know you found something, all right? It's not what you want to see, but you found something, all right? Then you move over the littlest bit to where your specimen should be, and you are pretty close to in focus when you found the wax, all right, and got it to look pretty clear, all right? At that point, you only use this fine focus knob, all right? You don't need the coarse adjustment because you are already right in the ballpark, right? And it's just focusing in on the detail, all right? And at this point, for every other lens that we flip into, it's only fine focusing, all right? The microscopes that we work with are called parfocal microscopes. And what that means is as we're looking through these ocular lenses and down through the objectives, once you find it in the lowest power, you've basically found it, right? And every other lens you turn into, that specimen is right, very close. It's probably not going to be there, crystal clear each time you switch lenses, but it is very close. And it's just a fine coarse or fine adjustment, not a fine coarse, a fine adjustment with that small knob one way or the other. I can't tell you which way to turn it because it really depends on where you're at with that specimen, but just a little bit of turning one way or the other, and it should bring it back into focus. Next lens, fine focus, till you get to the oil. And in between those two lenses, you put a drop of oil on the slide, put the oil immersion lens into it, fine focus, and you should have a nice crystal clear image, right? And then you can move it around with these knobs back and forth and see what's happening, all right? When I first am trying to find stuff under the microscope, my hand's on the coarse knob, my hand is also on the slide, on the knob to move it back and forth, and I wiggle the slide a little bit. All right. And if I'm looking through those oculars and I see stuff and I'm wiggling the slide back and forth and that stuff is not moving, I'm not seeing stuff on the slide. All right. I'm seeing something up on the oculars. There's dust. Sometimes your eyes are too close and you can see your eyelashes. Looks awesome. It's not what you want to see. All right. You need to back it up a little bit. All right. So if I start wiggling that slide back and forth the littlest bit and I'm looking through my oculars and it's moving too, I know I've found something, right? I'm not focused in on it yet, but I'm focusing in on the slide and I can see it moving. So I'm getting close to the ballpark, all right? And then I start a little slower moving it, trying to find it, all right? So the oculars up at the top where you're looking typically have a magnification of 10x. The objectives down below, it depends on the types of microscopes, but we think of from usually about 4x to give us 4 times 10, 40 magnification. all the way up to 100x on the oil immersion to give us 1,000 magnification, right? 10 times 100, right? And that gives us pretty much the best resolution we can get with this type of a microscope. You can bump it up a little bit more magnified. It doesn't really look any better, right? You can bump it down the littlest bit as well. Not much of a difference, right? Right around 1,000 is usually what we think about, right? When we hold these microscopes, the arm of the microscope back here, we will grab it back here and hold it by the base, 2 hands to carry it. Typically, there's a spot back here for the cord to wrap around. You also want to make sure that the stage is all of the way dropped down and the lowest power objective is in position so that when the microscope is put away, all of these lenses are kind of safe and protected. If the stage is up high and something happens and they get bumped, you may potentially cause damage to these oculars or these objectives, excuse me. And we want to try to minimize that. There's lens paper in the lab and also some lens cleaner that we can use to clean the oil off of the oil lens. The other lenses shouldn't be touching the oil in the first place. Usually the way you've got the stage and all of that stuff set up prevents that from ever happening in the first place. Okay. So here is a blow up with these lenses and our oil immersion lens. You can see this, when you first use it, you're going to be pretty stinking sure that you're going to break something. All right. That lens gets right up on that slide. All right. Merely touching it. All right. It is right here, this little bit of the lens, it is kind of spring loaded. So if you do zoom in, you're trying to focus and you get too close, it's going to push it up in there. You can technically crank it a bit and cause the slide to break. Usually it's the slide that gives first, right? You will know, right? You put a drop of oil on there and you can see it gets very close to the slide, but it doesn't actually touch it, right? And that oil allows all of the light to shine up into the lens versus a slide where no oil was applied, all right? And you can see we do get some refraction and reflection with that light as it bends and moves through there, all right? Not focusing or harnessing all of that light to give us a crisp image, right? And you definitely can tell, all right? If you are in there and you are trying to see something under oil immersion and you haven't put it on there, you can tell, right? Even if you are super close to the slide, you're not going to get a nice crisp image, right? You can tell that it's not on there, all right? You also can tell if you are staining something up and you can see something under the low power and you flip to the next lens and you can start to see something. And you flip to the third lens and you can see stuff pretty good. And then you put a drop of oil on there and you see nothing, right? And you focus one way and you focus another way and you still see nothing, all right? I can almost guarantee that what you have done is you've put your slide on upside down, all right? You can see those organisms when the slide's upside down. With the lower powered objectives, we're focusing in on the bacteria that's on the bottom of the slide, but we can still see it. But when we get to the oil bit, now the bacteria is down here and it should be up here. And that focal point isn't where we can see it, it's well below it. And you will never find it in focus, right? So if you are struggling, right? If you are not really knowing what's up with this, sometimes just flipping your slide over allows you to see it, right? Another trick can be with those wax pencils. You don't want to use a Sharpie because when we stain things, we use alcohol to decolorize, and alcohol and Sharpie will wipe right away, right? But a wax pencil does not. And if you draw with a wax pencil on one side of your slide and you're consistent, I always put an X on the top of my slide, right? And then I know. And then I can see it, right? And I know this side's up, right? And I can also move over to the X and focus on it first and go. Or I draw a circle around where I'm going to put my smear and my circle's on the top and I know it's always there. then I know and I can orient myself and I know what's up, all right? If you don't do anything like that and you just state stuff, sometimes there's issues, all right? Okay, guys, questions or concerns? Farewell, have a good day.